If you not subscribed, please subscribe the channel and click the bell button for more videos. Title, Class 11 Biology Chapter 9, Biomolecules. Teacher. Hello students. Welcome back to our biology class. Today we are going to study Class 11 Biology Chapter 9, Biomolecules. In the previous chapter, we studied the cell as the unit of life. But now we are going one level deeper. We are going inside the cell and asking, what is the cell made of? Just like a house is made of bricks, cement, iron, wood, and paint, a living cell is made of chemicals. These chemicals present in living organisms are called biomolecules. Today we will understand proteins, carbohydrates, lipids, nucleic acids, and enzymes in a very simple way with real-life examples. Part 1. What are biomolecules? Biomolecules are chemical compounds found in living organisms. Examples: carbohydrates, proteins, lipids, nucleic acids, amino acids, fatty acids, nucleotides, water, mineral ions. Teacher, students, are living organisms made of special elements that are not found in non-living things? Student: No, ma'am. Living and non-living things have many common elements. Teacher: Correct. Carbon, hydrogen, oxygen, nitrogen, and minerals are found in living organisms. But the important difference is their relative abundance. Living organisms have more carbon and hydrogen compared to non-living matter. Real-life example. A human body and soil both contain elements, but the arrangement and amount of these elements are different. That difference makes living systems special. Part 2, How to analyze chemical composition. To analyze living tissue, scientists take a sample like leaf, liver, or vegetable tissue and grind it with trichloroacetic acid using mortar and pestle. After filtering, two fractions are obtained. 1 acid-soluble pool. 2 acid-insoluble fraction. The acid-soluble pool contains small biomolecules. The acid-insoluble fraction contains larger molecules like proteins, nucleic acids, and polysaccharides. Real-life example. Think of making juice at home. When you filter the juice, the liquid passes through and the pulp remains. Similarly, in tissue analysis, some molecules come in the filtrate and some remain in the residue. Part 3, Organic and inorganic constituents. Living tissues contain organic and inorganic substances. Organic substances include amino acids, sugars, fatty acids, nucleotides, proteins, nucleic acids, polysaccharides, lipids. Inorganic substances include water, sodium ions, potassium ions, calcium ions, magnesium ions, chloride, phosphate, sulfate. Teacher, what is the most abundant chemical in living organisms? Student: Water. Teacher: Very good. Water makes up nearly 70 to 90% of cellular mass. Part 4, Amino acids. Amino acids are organic compounds that contain an amino group and a carboxyl group attached to the same alpha carbon. A general amino acid has amino group, carboxyl group, hydrogen atom, variable R group. The R group decides the type and property of the amino acid. Examples: glycine, alanine, serine, valine, lysine, glutamic acid. Amino acids are building blocks of proteins. Real-life example. Think of amino acids like different colored beads. When many beads are joined in a chain, they form a necklace. Similarly, amino acids join to form proteins. Question. Teacher, why are there different amino acids? Student: Because their R groups are different. Part 5, Types of amino acids. Based on properties, amino acids can be Acidic amino acids. Basic amino acids. Neutral amino acids. Aromatic amino acids. Examples. Glutamic acid is acidic. Lysine is basic. Valine is neutral. Tyrosine, phenylalanine, and tryptophan are aromatic. Amino acids may also exist in zwitterionic form depending on pH. Simple explanation. A zwitterion has both positive and negative charges in the same molecule. Part 6, Lipids. Lipids are generally insoluble in water. Examples. Fatty acids. Glycerol. Fats. Oils. Phospholipids. Cholesterol. A fatty acid has a carboxyl group attached to a hydrocarbon chain. Fatty acids can be saturated fatty acids, no double bonds. Unsaturated fatty acids, one or more double bonds. Fats and oils are formed when fatty acids combine with glycerol. Real-life example. Cooking oil does not mix with water because lipids are water insoluble. Teacher, where are phospholipids found? Student, in the cell membrane. Teacher, correct. Phospholipids are important components of biological membranes. Part 7, Sugars and carbohydrates. Sugars are carbohydrates. Examples. Glucose. Ribose. Fructose. Glucose is an important energy source. Ribose is present in RNA. Deoxyribose is present in DNA. Real-life example. When we eat rice, chapati, or potato, carbohydrates are digested and finally give glucose, which cells use for energy. Part 8, Nucleosides and nucleotides. Nitrogen bases include adenine, guanine, cytosine, thymine, uracil. When a nitrogen base combines with sugar, it forms a nucleoside. When phosphate is also added, it becomes a nucleotide. Nucleoside equals sugar plus nitrogen base. Nucleotide equals sugar plus nitrogen base plus phosphate. DNA and RNA are made of nucleotides. Question. Teacher, what is the building block of nucleic acids? Student, nucleotide. Part 9, Primary and Secondary Metabolites. Metabolites are biomolecules involved in metabolism. Primary metabolites are directly involved in normal growth and physiological processes. Examples. Amino acids. Sugars. Nucleotides. Fatty acids. Secondary metabolites are compounds mostly found in plants, fungi, and microbes. They may not be directly involved in basic growth, but many are useful. Examples. Alkaloids. Flavonoids. Rubber. Essential oils. Antibiotics. Pigments. Scents. Spices. Gums. Real-life example. Turmeric contains curcumin. Rubber comes from plants. Essential oils give fragrance. Many antibiotics are obtained from microbes. Part 10, Biomacromolecules. Biomolecules are of two types based on molecular weight. 1 micromolecules. These have molecular weight less than 1000 Daltons. Examples, amino acids, sugars, nucleotides, fatty acids. 2 macromolecules. These are large molecules. Examples, proteins, nucleic acids, polysaccharides. Lipids are small molecular weight molecules but appear in the acid-insoluble fraction because they are associated with membranes. Simple example. Small biomolecules are like individual bricks. Macromolecules are like big walls built using many bricks. Part 11, Average composition of cells. Average cellular composition. Water, 70 to 90%. Proteins, 10 to 15%. Nucleic acids, 5 to 7%. Carbohydrates, about 3%. Lipids, about 2%. Ions, about 1%. Teacher, which biomolecule is most abundant in cells? Student, water. Teacher, correct. Water is the most abundant chemical in living organisms. Part 12, Proteins. Proteins are polypeptides. They are linear chains of amino acids joined by peptide bonds. Each protein is a polymer of amino acids. Since proteins contain different kinds of amino acids, proteins are heteropolymers. Functions of proteins: structural support, enzyme action, hormonal function, transport, immunity, receptors. Examples: collagen gives structural support. Trypsin is an enzyme. Insulin is a hormone. Antibodies fight infection. GLUT4 helps glucose transport into cells. Real-life example: your muscles, skin, hair, enzymes, and antibodies all need proteins. Question: teacher, why should we eat protein-rich food? Student, because proteins provide essential amino acids needed for body growth and repair. Part 13, Most abundant proteins. Collagen is the most abundant protein in the animal world. RuBisCO is the most abundant protein in the whole biosphere. Real-life example: collagen is important in connective tissues. RuBisCO is present in plants and helps in photosynthesis. Part 14, Polysaccharides. Polysaccharides are long chains of sugars. Examples: cellulose, starch, glycogen, inulin, chitin. Cellulose is found in plant cell walls. Starch is stored food in plants. Glycogen is stored food in animals. Inulin is a polymer of fructose. Chitin is found in the exoskeleton of arthropods. Real-life example: paper and cotton contain cellulose. Potato contains starch. Our liver and muscles store glycogen. Insects have chitin in their outer covering. Part 15, Starch and iodine test. Starch forms helical secondary structures. Iodine molecules can fit into the helical structure of starch. So starch gives blue color with iodine. Cellulose does not form such complex helices and cannot hold iodine. Real-life example: in school lab, when iodine is added to starch solution, it turns blue-black. Part 16, Nucleic acids. Nucleic acids are polynucleotides. Two main types: DNA, deoxyribonucleic acid; RNA, ribonucleic acid. Each nucleotide has nitrogen base, sugar, phosphate. DNA contains deoxyribose sugar. RNA contains ribose sugar. DNA and RNA function as genetic material. Question: teacher, what is the function of DNA? Student, DNA stores genetic information. Part 17, Structure of proteins. Proteins have 4 levels of structure. 1 primary structure. This is the sequence of amino acids in a protein chain. 2 secondary structure. The protein chain folds into structures like alpha helix and beta pleated sheet. 3 tertiary structure. The protein folds into a 3-dimensional structure. 4 quaternary structure. More than one polypeptide chain or subunit comes together. Example: human hemoglobin has 4 subunits, 2 alpha and 2 beta chains. Real-life example: a thread can remain straight, form a spring, become a ball, or join with other balls to make a complex structure. Protein folding is similar. Part 18, Enzymes. Enzymes are biological catalysts. Almost all enzymes are proteins. Some RNA molecules also act like enzymes. These are called ribozymes. Enzymes speed up chemical reactions without being consumed. Real-life example: your saliva contains amylase, which helps digest starch. Teacher, why are enzymes important? Student, they make reactions happen faster inside the body. Part 19, Active Site. An enzyme has a special pocket called the active site. The substrate fits into the active site. The enzyme converts substrate into product. Substrate rightward arrow product. Real-life example. Think of lock and key. The enzyme is like a lock and the substrate is like a key. Only the correct substrate fits properly. Part 20, Chemical Reactions and Rate. Chemical reaction means bonds are broken and new bonds are formed. Rate of reaction means amount of product formed per unit time. Enzymes increase the rate of reactions. Example. Carbonic anhydrase converts carbon dioxide and water into carbonic acid very fast. Without enzyme, the reaction is slow. With enzyme, the reaction becomes extremely fast. Part 21, Metabolic Pathway. A metabolic pathway is a sequence of enzyme catalyzed reactions. Example. Glucose becomes pyruvic acid through several steps. Under anaerobic conditions in muscles, lactic acid is formed. In yeast fermentation, ethanol is formed. Real-life example. When we exercise heavily, muscles may form lactic acid, causing tiredness or pain. Part 22, Activation Energy. For any reaction to happen, substrate must pass through a high-energy transition state. The energy needed to reach the state is called activation energy. Enzymes lower the activation energy and make reactions easier. Real-life example. To push a heavy cycle over a speed breaker, you need effort. If the road is smooth, less effort is needed. Enzymes make the reaction path easier like a smooth road. Part 23, Nature of Enzyme Action. Enzyme action occurs in steps. 1. Substrate binds to the active site. 2. Enzyme changes shape and fits tightly around the substrate. 3. Bonds are broken or formed. 4. Product is released. 5. Enzyme becomes free to act again. Equation. E plus S rightward arrow E S rightward arrow E P rightward arrow E plus P. Part 24, Factors affecting enzyme activity. Enzyme activity is affected by temperature, pH, substrate concentration, inhibitors, temperature. Each enzyme has an optimum temperature. Low temperature makes enzyme temporarily inactive. High temperature denatures enzyme. pH. Each enzyme has an optimum pH. Too acidic or too alkaline conditions reduce activity. Substrate concentration. As substrate concentration increases, enzyme activity increases at first. After saturation, maximum velocity is reached. Inhibitors. Chemicals that reduce enzyme activity are called inhibitors. Part 25, Competitive inhibition. A competitive inhibitor resembles the substrate. It competes with substrate for the active site. Example. Malonate inhibits succinic dehydrogenase because it resembles succinate. Real-life example. Imagine two students trying to sit in the same chair. Only one can sit. Similarly, substrate and inhibitor compete for the active site. Part 26, Classification of enzymes. Enzymes are classified into 6 major groups. 1. Oxidoreductases. Catalase oxidation reduction reactions. 2. Transferases. Transfer functional groups from one molecule to another. 3. Hydrolyses. Catalase hydrolysis reactions. 4. Lyases. Remove groups without hydrolysis, often forming double bonds. 5. Isomerases. Convert one isomer into another. 6. Ligases. Join two compounds together using energy. Part 27, Cofactors. Some enzymes need non-protein helpers called cofactors. 3 types. 1. Prosthetic groups. 2. Coenzymes. 3. Metal ions. Prosthetic groups are tightly bound to enzymes. Example, HEM in catalase and peroxidase. Coenzymes bind temporarily during reaction. Many coenzymes contain vitamins. Example, NAD and NADP contain niacin. Metal ions help enzyme activity. Example, zinc is a cofactor for carboxypeptidase. Teacher, what happens if cofactor is removed? Student, enzyme loses catalytic activity. Final revision. Biomolecules are chemicals present in living organisms. Water is the most abundant chemical in cells. Amino acids form proteins. Fatty acids and glycerol form fats and oils. Nucleotides form DNA and RNA. Primary metabolites are essential for normal growth. Secondary metabolites include alkaloids, pigments, spices, drugs and rubber. Proteins are polypeptides made of amino acids. Polysaccharides are long chains of sugars. DNA and RNA are nucleic acids. Proteins have primary, secondary, tertiary, and quaternary structures. Enzymes are biological catalysts. Enzymes lower activation energy. Temperature, pH, substrate concentration, and inhibitors affect enzyme activity. Cofactors help some enzymes function properly. Q1. What are biomolecules? Biomolecules are chemical compounds found in living organisms. Q2. Name 4 major biomolecules. Proteins, carbohydrates, lipids and nucleic acids. Q3. What is the most abundant chemical in living organisms? Water is the most abundant chemical in living organisms. Q4. What is acid-soluble pool? The filtrate obtained after grinding living tissue in trichloroacetic acid and filtering is called the acid-soluble pool. Q5. What is acid-insoluble fraction? The residue or retentate left after filtration is called the acid-insoluble fraction. Q6. What are micromolecules? Biomolecules with molecular weight less than 1000 daltons are called micromolecules. Q7. What are macromolecules? Large biomolecules with molecular weight usually above 10,000 daltons are called macromolecules. Q8. Name the true biomacromolecules. Proteins, nucleic acids, and polysaccharides. Q9. Why are lipids found in acid-insoluble fraction? Because lipids are associated with cell membranes and form insoluble vesicles when tissues are disrupted. Q10. What are amino acids? Amino acids are organic compounds containing amino group and carboxyl group attached to the same alpha carbon. Q11. What is the general structure of amino acid? Amino group, carboxyl group, hydrogen atom, and variable R group attached to alpha carbon. Q12. How many amino acids occur in proteins? 20 amino acids occur in proteins. Q13. What is zwitterionic form? A molecule having both positive and negative charges is called zwitterionic form. Q14. What are lipids? Lipids are generally water-insoluble biomolecules such as fats, oils, phospholipids, and cholesterol. Q15. What are saturated fatty acids? Fatty acids without double bonds are called saturated fatty acids. Q16. What are unsaturated fatty acids? Fatty acids with one or more double bonds are called unsaturated fatty acids. Q17. What are phospholipids? Lipids containing phosphate group are called phospholipids. Q18. Where are phospholipids found? Phospholipids are found in cell membranes. Q19. What are nucleosides? A nitrogen base attached to a sugar is called a nucleoside. Q20. What are nucleotides? A nitrogen base, sugar, and phosphate together form a nucleotide. Q21. Name 5 nitrogen bases. Adenine, guanine, cytosine, thymine, and uracil. Q22. What are purines? Adenine and guanine are purines. Q23. What are pyrimidines? Cytosine, thymine, and uracil are pyrimidines. Q24. What is DNA? DNA is deoxyribonucleic acid and it contains deoxyribose sugar. Q25. What is RNA? RNA is ribonucleic acid and it contains ribose sugar. Q26. What are primary metabolites? Primary metabolites are biomolecules involved in normal growth and physiological processes. Q27. Give examples of primary metabolites. Amino acids, sugars, nucleotides and fatty acids. Q28. What are secondary metabolites? Secondary metabolites are compounds produced by plants, fungi, and microbes, often useful in defense, ecology, or human welfare. Q29. Give examples of secondary metabolites. Alkaloids, flavonoids, rubber, essential oils, antibiotics, pigments, spices and gums. Q30. What are proteins? Proteins are polypeptides made of amino acids linked by peptide bonds. Q31. Why are proteins called heteropolymers? Because proteins are made of different types of amino acids. Q32. What is collagen? Collagen is the most abundant protein in the animal world. Q33. What is RuBisCO? RuBisCO is the most abundant protein in the biosphere. Q34. What is a peptide bond? The bond joining two amino acids in a protein is called a peptide bond. Q35. What are polysaccharides? Polysaccharides are long chains of sugar molecules. Q36. Give examples of polysaccharides. Cellulose, starch, glycogen, inulin, and chitin. Q37. What is cellulose? Cellulose is a polysaccharide made of glucose and is found in plant cell walls. Q38. What is glycogen? Glycogen is a storage polysaccharide in animals. Q39. What is chitin? Chitin is a complex polysaccharide found in the exoskeleton of arthropods. Q40. Why does starch give blue color with iodine? Because iodine fits into the helical structure of starch. Q41. What are nucleic acids? Nucleic acids are polynucleotides like DNA and RNA. Q42. What is primary structure of protein? The sequence of amino acids in a protein is called primary structure. Q43. What is secondary structure of protein? Folding of protein chain into alpha helix or beta pleated sheet is called secondary structure. Q44. What is tertiary structure? Three-dimensional folding of a protein chain is called tertiary structure. Q45. What is quaternary structure? Arrangement of more than one polypeptide subunit forms quaternary structure. Q46. How many subunits are present in human hemoglobin? Human hemoglobin has 4 subunits, 2 alpha and 2 beta. Q47. What are enzymes? Enzymes are biological catalysts. Q48. What is active site? The pocket or crevice of an enzyme where substrate binds is called active site. Q49. What is substrate? The molecule on which enzyme acts is called substrate. Q50. What is activation energy? The energy needed to reach the transition state of a reaction is called activation energy. Q51. How do enzymes increase reaction rate? Enzymes lower activation energy and make reactions faster. Q52. What is ES complex? The enzyme substrate complex formed when substrate binds to enzyme is called ES complex. Q53. Write the enzyme action equation. E plus S rightward arrow ES rightward arrow EP rightward arrow E plus P. Q54. Name factors affecting enzyme activity. Temperature, pH, substrate concentration, and inhibitors. Q55. What is optimum temperature? The temperature at which enzyme activity is maximum is called optimum temperature. Q56. What happens to enzymes at high temperature? High temperature denatures enzymes and destroys their activity. Q57. What is competitive inhibition? Inhibition where inhibitor resembles substrate and competes for active site is called competitive inhibition. Q58. Give an example of competitive inhibition. Malonate inhibits succinic dehydrogenase by resembling succinate. Q59. Name 6 classes of enzymes. Oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Q60. What are cofactors? Non-protein components required by some enzymes for activity are called cofactors. Q61. Name 3 types of cofactors. Prosthetic groups, coenzymes, and metal ions. Q62. What is apoenzyme? The protein part of an enzyme without its cofactor is called apoenzyme. Q63. Give an example of prosthetic group. Heme catalase and peroxidase. Q64. Give an example of metal ion cofactor. Zinc in carboxypeptidase. Q65. What happens when cofactor is removed? The enzyme loses catalytic activity.
வணக்கம் மாணவர்களே. இன்று நாம் கிளாஸ் 11 பயாலஜி சாப்டர் 9 பயோமோலிக்யூல்ஸ் பற்றி எளிமையாக பார்க்க போகிறோம். முந்தைய பாடத்தில் நாம் செல் பற்றி படித்தோம். இப்போது அந்த செல் உள்ளே என்னென்ன வேதிப்பொருட்கள் இருக்கின்றன என்பதை பார்க்க போகிறோம். உயிருள்ள உடலில் இருக்கும் முக்கிய வேதிப்பொருட்கள் பயோமோலிக்யூல்ஸ் என்று அழைக்கப்படுகின்றன. கார்போஹைட்ரேட்ஸ், புரோட்டீன்ஸ், லிபிட்ஸ், நியூக்ளிக் ஆசிட்ஸ், வாட்டர், மினரல் அயான்ஸ் ஆகியவை பயோமோலிக்யூல்ஸ் ஆகும். கார்போஹைட்ரேட்ஸ் நம் உடலுக்கு எனர்ஜி தரும். நாம் சாப்பிடும் ரைஸ், சப்பாத்தி, பொட்டேட்டோ போன்ற உணவுகள் டைஜஸ்டன் ஆன பிறகு குளுக்கோஸ் ஆக மாறி செல்க்கு எனர்ஜி தருகின்றன. புரோட்டீன்ஸ் நம் உடலில் பல வேலைகளை செய்கின்றன. மசில், ஸ்கின், ஹேர், என்சைம்ஸ், ஆன்டிபாடிஸ் எல்லாவற்றுக்கும் புரோட்டீன்ஸ் முக்கியம். புரோட்டீன்ஸ் அமினோ ஆசிட்ஸ் என்ற சிறிய பில்டிங் பிளாக்ஸால் உருவாகின்றன. எப்படி சிறிய மணிகளை சேர்த்து மாலை செய்வோமோ அதுபோல் அமினோ ஆசிட்ஸ் சேர்ந்து புரோட்டீன் ஆகிறது. லிபிட்ஸ் தண்ணீரில் கரையாத பயோமோலிக்யூல்ஸ். ஆயில் தண்ணீரில் கலக்காது அல்லவா? அதுபோல லிபிட்ஸ் வாட்டர் இன்சொல்யூபிள் ஃபேட்ஸ், ஆயில்ஸ், கொலஸ்டரால், செல் மெம்பரேனில் இருக்கும் பாஸ்போலிபிட் போன்றவை லிபிட்ஸ். நியூக்ளிக் ஆசிட்ஸ் என்பது டிஎன்ஏ மற்றும் ஆர்என்ஏ. டிஎன்ஏ நம் உடலின் ஜெனடிக் இன்ஃபர்மேஷன் ஐ சேமித்து வைத்திருக்கும். இது ஒரு இன்ஸ்ட்ரக்ஷன் புக் மாதிரி வேலை செய்கிறது. டிஎன்ஏ மற்றும் ஆர்என்ஏ நியூக்ளியோடைடு என்ற சிறிய யூனிட்ஸால் உருவாகின்றன. ஒரு நியூக்ளியோடைடு சுகர், நைட்ரோஜன் பேஸ் மற்றும் டிஎன்ஏ ஆர்என்ஏ அமைப்பை உருவாக்க உதவும் ஒரு கெமிக்கல் குரூப் இருக்கும். பிரைமரி மெட்டாபோலைட்ஸ் என்பது உயிரின் அடிப்படை செயல்பாடுகளுக்கு தேவையான பயோமோலிக்யூல்ஸ். அமினோ ஆசிட்ஸ், சுகர்ஸ், நியூக்ளியோடைட்ஸ், ஃபேட்டி ஆசிட்ஸ் போன்றவை பிரைமரி மெட்டாபோலைட்ஸ். செகண்டரி மெட்டாபோலைட்ஸ் என்பது பிளான்ட்ஸ், ஃபங்கி, மைக்ரோபீஸ் போன்றவற்றில் காணப்படும் சிறப்பு பொருட்கள். ரப்பர், ஸ்பைசஸ், சென்ட்ஸ், பிக்மெண்ட்ஸ், ஆன்டிபயோட்டிக்ஸ், டர்மரிக்கில் இருக்கும் கர்குமின் போன்றவை செகண்டரி மெட்டாபோலைட்ஸ். இவை மனிதர்களுக்கு மிகவும் பயனுள்ளவை. பயோமக்ரோமோலிக்யூல்ஸ் என்பது பெரிய மோலிக்யூல்ஸ். புரோட்டீன்ஸ், நியூக்ளிக் ஆசிட்ஸ், பாலிசாக்கரைட்ஸ் ஆகியவை முக்கிய பயோமக்ரோமோலிக்யூல்ஸ். பாலிசாக்கரைட்ஸ் என்பது நீளமான சுகர் செயின்ஸ். செல்லுலோஸ் பிளான்ட்ஸ் செல் வால் இருக்கும். ஸ்டார்ச் பிளான்ட்ஸில் ஸ்டோர்ட் ஃபுட் ஆக இருக்கும். கிளைகோஜன் அனிமல்ஸில் ஸ்டோர்ட் ஃபுட் ஆக இருக்கும். ஜெட்டின் இன்செக்ட்ஸ் அவுட்டர் கவரிங் இருக்கும். என்சைம்ஸ் என்பது பயோலாஜிக்கல் கேட்டலிஸ்ட்ஸ். உடலில் கெமிக்கல் ரியாக்ஷன்ஸ் வேகமாக நடக்க என்சைம்ஸ் உதவுகின்றன. எடுத்துக்காட்டு, நம் சலைவா தில் அமிலேஸ் என்சைம் இருக்கும். அது ஸ்டார்ச் டைஜஸ்டனுக்கு உதவும். என்சைம் தி ஆக்டிவ் சைட் என்ற ஒரு சிறிய பாக்கெட் இருக்கும். சப்ஸ்ட்ரேட் அந்த பாக்கெட்டில் ஃபிட் ஆகும் பிறகு என்சைம் சப்ஸ்ட்ரேட் ஐ ப்ராடக்டாக மாற்றும். இதை கிளாக் அண்ட் கீ மாடல் போல நினைவில் வைத்துக் கொள்ளலாம். என்சைம் லாக் மாதிரி, சப்ஸ்ட்ரேட் கீ மாதிரி. என்சைம்ஸ் ஆக்டிவேஷன் எனர்ஜி ஐ குறித்து ரியாக்ஷன் வேகமாக நடக்க உதவுகின்றன. என்சைம் ஆக்டிவிட்டி டெம்பரேச்சர், pH, சப்ஸ்ட்ரேட் கான்சென்ட்ரேஷன், இன்ஹிபிட்டர்ஸ் ஆகியவற்றால் பாதிக்கப்படும். அதிக டெம்பரேச்சர் என்சைம் ஐ டேமேஜ் செய்யும். ஒவ்வொரு என்சைம் ஒரு ஆப்டிமம் டெம்பரேச்சர் மற்றும் ஆப்டிமம் pH இருக்கும். இப்போது சாப்டர் 9 ஐ சிம்பிளா ரிவைஸ் பண்ணுவோம். கார்போஹைட்ரேட்ஸ் எனர்ஜி தரும், புரோட்டீன்ஸ் பாடி ஒர்க் மற்றும் ரிப்பேருக்கு உதவும், லிபிட் செல் மெம்பரேன் உருவாக்கும் மற்றும் எனர்ஜி ஸ்டோர் செய்யும், நியூக்ளிக் ஆசிட்ஸ் ஜெனடிக் இன்ஃபர்மேஷன் ஸ்டோர் செய்யும், என்சைம்ஸ் கெமிக்கல் ரியாக்ஷன்ஸ் ஐ வேகப்படுத்தும். பயோமோலிக்யூல்ஸ் சாப்டர் ஐ மனப்பாடம் செய்யாமல் வாழ்க்கையுடன் கனெக்ட் செய்து புரிந்து கொள்ளுங்கள். நாம் சாப்பிடும் உணவு நம் உடலின் எனர்ஜி, நம் DNA இன்ஃபர்மேஷன், நம் டைஜஸ்டன், நம் growth எல்லாமே பயோமோலிக்யூல்ஸ் ஐ சார்ந்தது. இதுதான் பயோமோலிக்யூல்ஸ் என்ற உயிரின் வேதியியல் உலகம். நன்றி மாணவர்களே, அடுத்த பாடத்தில் மீண்டும் சந்திப்போம்.
Dear students, if you like simple study videos, chapter explanations, question and answer practice, and easy revision notes, please subscribe to our channel, save this playlist, and continue learning step by step. Your support motivates us to create more useful study-related videos. Thank you for watching, and keep learning.